introduction on behalf of Passage Bio. I am honored to kick off the session. I am Samiah Al-Zaidy. I am a pediatric neurologist, and I'm leading the development of the Krabbe program here at Passage. So my goal today is to share an overview of our phase 1/2 clinical trial, where we are assessing the safety and efficacy of an investigational treatment, which I will be referring to as PBKR03 throughout this presentation. And we are studying it in early infantile Krabbe disease. Next, please. So just a disclosure that PBKR03 is an investigational treatment. It is not approved by any of the regulatory agencies. And that in this study, we are assessing the safety and efficacy. Next, please. So at Passage, we do recognize the devastating nature of several lysosomal storage diseases, specifically some of the leukodystrophy and neurodegenerative disorders. Krabbe is one of 3 of the lysosomal storage disorders that we are currently studying. And this audience is no stranger to how devastating and rapidly progressive this disease is. And although I do have the incidence reference here as 2.6 per 100,000 live births worldwide, from what has been published, we definitely heard and learned a lot more yesterday from several presentations on newborn screenings from the states that are currently carrying out the newborn screening programs. Next, please. So one of the things we heard, it was an elegant presentation by Dr. Matern yesterday about examples of subjects who are identified in a day-by-day, how from the identification to the referral to treatment. And perhaps in the scientific literature, there's nothing more impactful than survival graphs that illustrate the urgent need and the medical urgency for early intervention. And as you can see here from Dr. Escalar's group, 70% of our patients would have passed away by the age of 2.5 years of age. So we do understand the diagnostic dilemma and that the early presentation of irritability and feeding difficulties it may result in delay in diagnosis, and this could impact the advancement of clinical therapies. Next, please. However, thank you to the families, the advocacy groups, and key opinion leaders who've been advocating for newborn screening. We did enjoy hearing about the progress that has been made. And while this is work in progress, both here in the United States as well as globally, we do believe that this early intervention will enable access to viable treatment options for these families, for which I will be discussing one of the treatment options that we are currently investigating at Passage Bio. Next, please. So to provide a very brief overview and in 15 minutes to cover the clinical trial, but also to give a background on AAV gene therapy can be somewhat challenging, but I will do my best. We utilize adeno-associated virus. This is a virus that has an excellent safety profile, has been utilized in several gene therapy studies in both the pediatric and adult populations. I personally have used it in several delivery methods with different serotypes. The capsid, or what we call the serotype that we use here, is AAV-Hu68. This is a variant of AAV9. And the beauty of serotype 9 is its excellent CNS biodistribution, whether you deliver it through systemic delivery through an intravenous injection or through an intrathecal, and particularly through a cisterna magna delivery, where you increase your CNS biodistribution. So the goal is that once this is delivered to the patient, it would serve as a shuttle or a vector to carry a fully functional human GALC gene, the gene that is defected in our children with infantile Krabbe disease. Now, the hope and what the theory has been for AAV is that once it is delivered, it is taken up by the cell, which we call transduction of the cell, and then the capsid delivers the transgene to the nucleus. And once the transgene is in the nucleus, it sits there as in an episomal fashion where it eventually produces a protein. Now, just with most enzyme therapies that utilize AAV gene therapy, these are one-time treatments for where we are in the field of gene therapy. The hope is that with this one-time treatment, if some cells are taking up the AAV gene therapy, that you would have the opportunity for what we call cross correction. And this is where if some cells are taking up the AAV gene therapy, they will produce the protein where the cell itself will benefit from it, but it'll also release the enzymes so that neighboring cells who may have not been transduced as efficiently as that cell would benefit from that. So that was the goal, and I'll demonstrate in the next slides in the animal studies that cross-correction did indeed improve behavioral and cognitive function in the murine models of several lysosomal storage diseases, but here in particular in the Krabbe animal models. Next, please. So how is PBKR03 administered in our current trial or the GALC trial? We utilize the approach of delivering it directly into the cerebrospinal fluid. So we are passing the blood-brain barrier by delivering it directly to the CSF through an intracisterna magna injection. So it is an intrathecal where you are puncturing the membranes and releasing the product into the cerebrospinal fluid. What's the advantage of that? You do have excellent CNS biodistribution. Once again, this is an AV9 serotype, so that's an advantage. When you bypass the systemic circulation, you do not require as high of viral vector loads as you do with systemic circulation. So it allows you to use lower doses with a localized injection into the cerebrospinal fluid. You also have the advantage of bypassing the impact of neutralizing antibodies on gene therapy. So AAV is something that many of us are exposed to. The prevalence of the antibodies that we do develop to the virus varies, and it increases with age. So while infants may have a lower prevalence of antibodies, it still remains a risk in preventing, in some clinical trials, children to receive gene therapy if they do have high titers of antibodies. With intrathecal delivery, particularly with cisterna magna delivery, the presence of neutralizing antibodies does not preclude these patients from receiving intracisterna magna injection. So we do administer this under sedation, utilizing modern neuroimaging to allow for precise delivery with a standardized protocol and procedures that are certified and trained to follow a specific protocol for standardization. We do know that this intracisterna magna, for those who have attended some of the recent symposia, many sponsors and many gene therapy trials have been utilizing this approach for both pediatric and adult patients. Next, please. So as most clinical trials, you start by assessing your investigational product and looking at your proof of concept studies in the mice. And in this spontaneous model of a twitcher mice, what we did see is that as you increase the dose, you do see a dose-dependent survival and motor function response. Now, these beautiful graphs were generated by Dr. Jim Wilson's lab, led by Juliette Hardot, who we are partnering with for our current products, and specifically here for our Krabbe product. And to your left, what you can see is the red line is where a vehicle was delivered. So it was saline, and you can see the median survival for an untreated twitcher mouse is 43 days. And as you increase the dose, you see a dose-dependent response with increased survival up to 130 days with that final high dose. And corresponding to that, we do see an improvement in motor function that is demonstrated by improvement in performance on the rotor rod test for these mice. Next, please. So one of the questions that it comes up is during the, as we discuss intracisterna magna delivery, what is evidence to support that you might have cross-correction or you might have an opportunity to preserve peripheral nerve function and health or restore myelin. And this is a great demonstration of improved GALC activity both in the brain, in the mice who received 2.2 × 10^11 genome copy after symptom onset. So these are juvenile mice who received this. And you can see that we see improvement in the GALC activity in the brain, but we also see it peripherally in the serum as well as in the liver. Now, to your right, and I'll walk you through each of these nerve sections, we're looking at peripheral nerve. How does PBKR03 impact the health of the peripheral nerve? So the top sciatic nerve is the sciatic nerve of a healthy mouse, and what you see in the blue is the staining for myelin. Now the middle is a twitcher mouse, so it is a untreated twitcher mouse that received vehicle treatment without a healthy transgene. And to the bottom, you can see a twitcher mice that is treated with PBKR03, and you can see that myelin has been preserved in these mice. Next, please. Now, moving on to a larger animal model, what we look at in the Krabbe dog model, we see that 6 months post-treatment, we continue to see that the CSF GALC activity has been maintained, as well as we do see a significant reduction in the CSF cytosine levels. Next, please. So how did this translate clinically in a large animal model? And in this Krabbe dog model, if you can click once forward the video on the left, please. So this is a Krabbe dog that did not receive treatment. As you can see, it is struggling to keep up, and you can see the hind limbs, the weakness in the hind limbs, and eventually these dogs usually pass away around 12 weeks of age. That's the median survival. And then if you can please click next, we see a Krabbe dog that did receive PBKR03, and as you can see, the dog is running around and playing with the instructor and with no evidence of weakness. Next, please. So these animal studies and the data from that has enabled an IND for a clinical trial to launch the phase 1/2 study delivering PBKR03. Now, this is a phase 1/2. It is a global study, so we do have sites in the United States as well as outside of the United States, so it's a multi-center. It is an open label. It is a single arm study and a dose escalation study. So the initial part will be a phase 1/2 dose escalation, followed by a confirmatory study in patients with early infantile Krabbe disease. The age group is from 1 to 9 months of age, and the division of the cohorts is based on older infants and younger infants, and this is largely a matter of testing the ICM procedure initially in older infants before proceeding to younger infants. So the low dose will be tested in the first cohort of 4 to 9 months of age. Each cohort was originally designed to enroll 3 subjects with the possibility to add an additional subject to each cohort in the event we had a serious adverse event. Now, this is a single dose that is delivered. The duration, the initial assessment will be at 2 years post-treatment, followed by an additional 3 years of long-term follow-up for safety and durability. Now, I'll walk you through the endpoints, but as you can see, cohort 1, we have expanded the cohort. We did enroll a first subject, and as we expanded, as we observed the patient, we did have a serious adverse event, and that triggered an increase in cohort 1. So we are expanding it to enroll 4 patients in this first cohort. Before moving on to the high dose in the older infants and the lower dose in the younger infants. And following that, we would be moving on to the high dose in the younger infants, and we will have a IDMC or a safety review of our data before moving on to our confirmatory cohort. Next, please. So our inclusion criteria, 1 to 9 months of age at enrollment. We do accept pre-symptomatic infants and those with symptomatic onset at less than 6 months of age, leukocyte GALC activity below the lower limit of normal, and whole blood cytosine above 10 in an effort to ensure that we are enrolling infantile Krabbe disease. We do look at pathogenic mutations, and if in the absence of 2 pathogenic mutations, we do look at the totality of the presentation, and the patient will go through an adjudication process to ensure that this is a subject who is predicted to develop infantile Krabbe disease. And symptomatic subjects do have to exhibit a minimum level of neurological and developmental function when age appropriate, and some of those include movement in the lower extremities, being able to support their head, reciprocal spinal. Next, please. So study outcomes, this is a phase 1/2 study. So our primary outcome obviously is safety. We are looking at treatment-related adverse events and serious adverse events that would be classified at a grade 3 or higher within 24 months of dosing. We also look at change from baseline in nerve conduction studies. With all AAV studies, we are familiar with dorsal root ganglion toxicity that has been reported in some of the animal studies with AAV gene therapy. So that is part of our safety assessments, but it's also an efficacy outcome measure for us as well. We're looking at safety laboratory assessments, and from an immune response standpoint, we are looking at both T cell and antibody response to the capsid, but as well to the transgene, depending on in both serum and CSF. As for the secondary outcomes, we're looking for clinically meaningful outcomes in the developmental milestones measured by Bailey and the Vineland. Our biomarkers, we are looking at GALC activity in blood and CSF. We are looking at cytosine in both blood and CSF, and we're looking at markers for disease progression as well. We have MRI studies that are being conducted as part of this study. We look at ventilator-free survival and feeding tube placement and quality of life, as most clinical trials are now including. Next, please. So I do want to make sure that we highlight that we are actively recruiting for our sites. The sites that are highlighted in green, including, I see Dr. Malak is on the call and will be speaking shortly. We do have these sites active and up and running. As you can see, we have sites here in the United States and North America, including Canada. And outside the US, we do have our site in the Netherlands that is up and running, as well as the UK and Israel. And we look forward to activating our site at Children's Hospital Philadelphia fairly soon, as well as Dr. Bonkowski and our site at Brazil. Next, please. So earlier this year, we did publicly disclose that our first patient has been enrolled and was dosed in the GALC trial. Next, please. So bringing this therapy would not have been possible if not for the tremendous efforts and collaborations throughout the study. On behalf of the team at Passage, I want to thank the patients, the families, the caregivers, and of course our wonderful clinical investigators who are advocating for their patients and the families, the key expert leaders who have contributed, and last but certainly not least, our partners at the University of Pennsylvania, led by Dr. Jim Wilson. And I do believe I'm at 1:32, so I will thank you all. And next slide, please feel free to reach out for any questions. I'll be happy to take questions during the discussion session or whenever Dr. Stone allows us to. Thank you.